All yeast flocculate to some extent, and at some point after attenuation. Lectins are thought to be primarily responsible for this trait. Lectins are carbohydrate-binding proteins. In this case, the lectins are presented at the yeast cell surface and interact with mannose-based oligosaccharides also presented on that surface. The lectin and oligosaccharide interact, forming a raft or flocculate. Generally, flocculation is associated with glucose depletion. Ultimately, strong flocculating yeast will settle to the bottom of the fermentation vessel, where they can be drawn off in a conical fermenter. In this image, we see that the non-flocculant culture attenuates, but doesn't fall out of solution to any great extent, whereas the highly flocculant culture more or less completely falls out after about 90% attenuation. The flocculation behavior of yeast strains varies widely, as do their responses to different environmental conditions. Let's take a closer look at one lectin, mannose-binding lectin. Sometimes these lectins are referred to as zymolectins, but they are C-type lectins, that is, calcium-dependent proteins. Mannose-binding protein mediates interactions between the protein and the mannose's 3' and 4' hydroxyls, as shown in this close-up on the right. This is the same configuration as glucose for these particular hydroxyls, and in fact, mannose-binding lectin does bind glucose and a range of other carbohydrates with lower affinity. Glucose's 2' equatorial hydroxyl decreases mannose-binding lectin's affinity relative to mannose. So I'm really showing you this image to show you the details of how calcium mediates the detailed interaction with mannose. The point is that mannose-binding lectins mediate cell-cell adhesion via mannose polymers presented at the yeast cell surface. Expression of mannose-binding lectins depends on calcium concentrations, nutritional conditions, metabolic state, and other factors. But it has been observed that calcium in the medium is depleted at the end of fermentation, at the end of attenuation, and the calcium accumulates at the cell wall. In some strains, however, flocculation appears to be less sensitive to calcium and mannose concentrations. The flocculation proteins are encoded by the flocculation genes, or the flow genes, and control of their expression is poorly understood. First of all, it's complex and responds to nutrient conditions, to pH, to temperature, and second, it's controlled epigenetically as well as genetically, meaning that remodeling of the genome of the parent affects expression patterns and flocculation characteristics of later generations. Thus, strains can change behaviors in the brewhouse depending on conditions. Here again, we see the yeast cell expressing the mannose binding proteins, which can be calcium activated so that they can then interact with the mannose oligosaccharides presented at the cell surface. Here again, just noting that effectors of flocculation characteristics are diverse, and therefore the process of flocculation itself is very complex and again varies among strains and within strains depending on conditions. Cell wall composition matures as cells are harvested. The more flocculent cells accumulate at the bottom of the vessel. Usually, yeast can be recycled over 7 to 10 generations before senescence and death. In this image, we see bud scars that reflect the number of daughters each cell has budded. In this electron micrograph, we see a couple of cases where the bud scar is apparent during fission. And here in the top right, we see a bud occurring before the scar has actually formed around it. A general takeaway is that membrane composition of the yeast changes with time and reproduction. The bud scars represent this. In a general way, then, if membrane composition and chemical characteristics are relevant to flocculation, then yeast flocculation characteristics are changeable as well. I've already suggested that selection of traits, either on purpose or by accident, happens in the brew house, even in the decision to harvest yeast from the krausen or from the trub. Lager is an excellent example of how yeast are shaped by the brewer and even the broader culture in which the brewery is situated. Recall that we usually refer to Saccharomyces cerevisiae when talking about brewing yeast, but this is the ale yeast, and the bottom fermenting lager yeast is adapted to the cold conditions in which the fermentation takes place. The species is known as Saccharomyces pastorianus, formerly referred to as Saccharomyces carlsbergensis, as it was isolated in the Carlsberg Brewery in Copenhagen by Emil Christensen. He's a remarkable figure, as he, in 1883, was the first to isolate a single colony of yeast, pioneering serial dilution to isolate single cells. Sulfate import is key to the function of Pastorianus. Upon import into the cell, sulfate displaces the two terminal phosphate moieties of ATP to form adenylyl phosphate sulfate, or APS. APS reductase takes that new structure and removes sulfate as sulfite. Sulfite, of course, is an important flavor stabilizing molecule and thus an important characteristic of lager yeasts. Here I'm showing you the reaction mechanism of APS reductase. Really, it's just its trigonal bipyramidal intermediate, where APS reductase has an FAD cofactor that is in a covalent complex with APS. So here's the sulfate moiety. The removal of sulfate's fourth oxygen is key here. Here you see the collapse of the intermediate occurs with the cleavage of this bond between the sulfur and the adenylyl phosphate's terminal oxygen, acts as leaving group. I should point out, too, that APS reductase is not a dehydrogenase like ADH, but both are oxidoreductases that use organic cofactors to carry out redox chemistry. APS reductase uses FADH, that's flavin adenine dinucleotide, instead of NADH, that is nicotine adenine dinucleotide, as the reducing cofactor. But back to fermentation. For sulfate to be converted to sulfite, it must be imported efficiently into the cell. Pastorianus is far more efficient as a sulfate importer than Cerevisiae. Where did this trait and the equally important cryotolerance come from? Well, given the long history of beer in Europe, we might think it has European origins, but in fact, it's part of the Columbian exchange, a hybrid between a Cerevisiae species and a New World strain. Researchers collected colonies from Patagonian trees and deduced that Pastorianus is an amalgam of Cerevisiae and Eubayanus, isolated from beech trees in Patagonia. Perhaps barrels made there made their way back to use in New World breweries, bringing New World yeast with it. It's profound to realize that something generally considered fundamentally European like lager actually wouldn't exist without hybridization with yeast from the American continent. The Kex2-dependent cryotolerance is also a Saccharomyces eubayanus donation, which allows the yeast to ferment efficiently at low temperatures, e.g., lager fermentation temperatures. The sulfate import is a complex story, but in the end, Pastorianus wound up with two highly active copies of Sol2, accounting for a lager-type beer that's high in sulfite-dependent microbial and flavor stability. As an example of the diversity of yeast strains, let's take a look at the White Labs catalog. Their strains of Saccharomyces cerevisiae are organized between 001 and 799. Belgians and Saisons, with their strong ester and phenol production, are set apart from other Saccharomyces cerevisiae strains in the 300s and 500s. Lagers are a different species of Saccharomyces and have numbers in the 800s. Note the cryotolerance in the Pilsner lager attributes shown here, and recall that Kexin-dependent cryotolerance was conferred on Pastorianus by its New World parent. Brett, Brettanomyces, though it's also a yeast, is categorized with the lactic acid bacteria and Pediococcus in the 600s.